Hello everyone. Our next module is about the cell structure and its function. So, there are two types of cells, that is the animal cell and the plant cells. So, this both cells contains what? The nucleus, cytoplasm, cell membrane, mitochondria. But plant cell actually contains what? The cell wall and chloroplast and vacuoles. So what is cell? So cell is a basic structural and functional unit of all living organism. All the living organism can be categorized into unicellular, that is protozoa, yeast and bacteria, and multicellular, that is the plants and animals. So it is first discovered by whom? The Robert Hooke, who named the cells from the slice of cork in 1665. Some organism, as I have told, that it can be say an unicellular organism or multicellular organism, and the diameter is approximately 2 to 120 micrometer. So these are the parts of the cell. Firstly, the mitochondria. So this mitochondria actually produces what? The energy for the cells to use to break down the substances during the oxidative metabolism. Then comes the cell membrane, that is made up of a double layer of the fatty material. It actually allows the materials to transfer into and out of the cell. It allows the food and the water to pass into the cell and waste to pass out of the cell. Then comes the cytoplasm. So the cytoplasm is actually a fluid material or a jelly-like substance that is present between the cell membrane and the nucleus. So this is where all the organelles of the cells are situated. Then comes the lysosome. Lysosome actually contains what? A enzyme known as lysozyme, where the digestion of the cell and the waste products are occurs. Then comes the endoplasmic reticulum. It actually divided into smooth endoplasmic reticulum and rough endoplasmic reticulum that store materials made by the cell. Then comes the DNA. DNA actually helps in coding information that passes on every generation characteristics. Next is the nucleus. This nucleus actually contains all the DNA materials, and this helps in controlling the cell for further division. Next is the Golgi complex, which collects the packages and distributes the molecules made in the cell. Next is the ribosome, that is a tiny protein producing factories of the proteins that helps in the replications of the cells. Then comes the vacuole. Vacuole actually are the storage area where the fats and other substances can be stored. Next is the peroxisome, that is a vesicle-like structure that contains enzymes that carry out particular reactions such as detoxifying of a harmful molecules. And lastly is the centriole. These are the tiny organs that actually helps in the cell division, that is the mitosis and the meiosis. So the cell theory was actually proposed by the Matthias Kelden and the Theodor Schwann in 1839. They actually explained that the all living organisms are made up of cells, which are actually the vital element for working of a unit life of a living organism. All cells come from the pre-existing cells through cell division. So what are the constituents, or with which type of materials these cells are made up of? Firstly, it contains what? The protoplasm. So this protoplasm actually composed of water, that it contains 70 to 80% of water. Then the carbohydrates, lipids, proteins, and electrolyte, that is sodium, potassium, magnesium, calcium, phosphate, chloride, and bicarbonate. So, what are the structure present in the cell? It is mainly constituted with the three part, that is cell membrane, cytoplasm and its organelles, and the nucleus. Firstly, we come to the cell membrane. So, the cell membrane is also known as the plasma membrane. So, this cell membrane is selectively permeable biological membrane which allows only certain substances to pass through it. It acts as a barrier between the inner and the outer surface of the cell. It is also known as the plasma membrane or the plasma lemma. It actually comprises of what? Many substances like proteins and lipids, along with the other living molecules. These molecules are important for the normal functioning of the cells. Example in an ion channel conductance and cell addition and cell signaling. So what are the properties of the cell membrane? It actually helps in what? The guarding of the cell against the external environment, right? So this is the structure of a fluid mosaic model. So this fluid mosaic model was given by the S.J. Singer in the University of California and G.L. Nicholson in 1970s. So according to this model, proteins do not form a continuous layer extending from one side of the membrane to the other. Rather, it is present in the form of globules dotted randomly in the mosaic pattern. So you can see this is the mosaic pattern and it is actually denoted in a dotted form. So some globules of the proteins penetrate into the membrane. So these are the proteins like the globular protein, glycoprotein. These are the protein channels, then the hydrophilic heads. These are the phospholipid bilayer. That is, it contains what? The polar head and the non-polar head. So some of the globules of the protein penetrate into the membrane to deeper extent. Some of them actually remain attached to the membrane surface. So some of these are remain attached to the membrane surface, while some globules extend through it and adhere to the other side. So it is extending in the outer surface to the inner surface. So it actually explains the rigidity of the cell membrane. Experiments on viscosity of the membrane show that it has a fluid-like consistency. This fluidity allows movement of the lipid and the protein molecules within it due to the fluidity and the mosaic arrangement of the protein molecules. This model is known as the fluid mosaic model. So it consists of what? The bilipid layer and the proteins that are integral proteins and the peripheral proteins. Then what is the function of the cell membrane? So it acts as a protective function. It provides digestion. It actually helps in selective permeability of the molecules. It is having a polar molecules and chemical and physical properties. So in the protective forms, it forms the outermost boundary of the cell organelles. Then in the digestive function, it actually takes the food and excretes out any waste debris from inside to the outside of the cell. Then comes the selective permeability. So it contains what? A polar head and a non-polar head. So these polar molecules, that is non-polar molecules, that is the oxygen, carbon dioxide, and nitrogen, lipids, steroid, hormones, alcohols, can dissolve through this non-polar region of the plasma membrane and can easily pass across the cell membrane. Then comes the polar molecules. As the plasma membrane contains the polar head, so these polar molecules that are soluble in the polar head region like the H2O soluble molecule, glucose, urea, etc., have much lower solubility, therefore penetrate the membrane much more slowly. Then comes the chemical and physical properties of the membrane, which control the free passage of the ions in and out of the cell. So this property helps in maintaining the components in ICF and ECF. Then comes the insulating properties. So it is having a dielectric property of a charged condenser. Thus the cell membrane have a very high insulating value. And that's why it actually can control the temperature of our body. Next part is the cytoplasm and its organelles. So this is the cytoplasm. So what I have told you earlier, that cytoplasm are the semi-fluid or jelly-like fluid which is present inside the cell of both the plant and animal. So this constitute the parts of cytoplasm are the cytosol, cell organelles, and cytoplasmic inclusions. They are having, or they are protected by the plasma membrane. So they are protected by the plasma membrane that contains the organelles in a eukaryotic cell. So what is cytosol? So the cytosol are the aqueous form or the part of the cytoplasm which is present outside all the organelles. It actually accounts for almost 70% of the total cell volume. Then comes the gelatinous substance consisting mainly of cytoskeleton, filaments, organic molecules, salt, and water. Chemically, cytoplasmic matrix is composed of atoms, ions, and molecules. Then comes the organelles. Organelles are present in the cytoplasm are explained here. That is firstly mitochondria, then endoplasmic reticulum, third is the lysosomes, fourth is the Golgi apparatus, fifth is peroxisomes, and sixth is vacuoles. So is this each organelles is actually protected by a lipid membrane that has a specific property of functions. So firstly comes the mitochondria. So mitochondria is actually commonly known as what? The powerhouse of the cell, because they generate what? The adenosine triphosphate, that is the ATP. So this ATP actually helps in the formation of the chemical energy. So this mitochondria thus are known as the chief source of the energy in the cell. So this energy actually released by what? The oxidation of the food during the cellular respiration, converted into highly energetic phosphate bond, ATP and ADP. This ATP bounded energy is utilized in different anabolic and catabolic processes, such as transport of ions across the cell membrane and muscle contraction or the synthesis of protein. So it was first observed by Colliker in 1850 as a granular structure in the striated muscle. So what is this dimension? The length is generally 5 to 12 micrometer and diameter is 0.5 to 1 micrometer. And it is in the shape of a globular shape or the filamentous shape. So here in the diagram we can see it contains an inner membrane. This one is the inner membrane and this is the outer membrane, and this folding like structure is actually known as the cristae, and these are the matrix present inside the cristae. So the components of the mitochondria are the outer membrane, inner membrane, and intermediate space. So this intermediate space is what? A space between an outer and the inner membrane. So cristae infoldings of inner membrane, that is cristae is the infolding of the inner membrane, and this cristae actually contains what? A matrix, a space enclosed by the inner membrane. This membrane are made up of what? The phospholipids and the proteins. Then comes the outermost membrane. Outermost membrane are consisting of a porins, a protein-like structure. These porins are a passage-like structure that forms a channel that allows what the molecules of size that is 5000 Dalton or less to pass. Then comes the interior part, that is the matrix part of the mitochondria. So it contains the enzymes concerned with what the citric acid cycle or the respiratory chain oxidation. So these are the major metabolic pathways which actually involved in the oxidation process of the carbohydrates, amino acids, lipids, and the biosynthetic pathways involving the heme synthesis, urea, which are located in the inner matrix. So this inner membrane, as I have told you earlier, contains what the ATPase enzyme which and the other enzyme which helps in the synthesis of what the ATP and metabolism of the ATP. This ultimate purpose of this mechanism is what the oxidative phosphorylation and synthesis of ATP. Mitochondria has some protein synthesized by the mitochondrial DNA. Then comes the functions. So what is the function? As a common function is what? It is a powerhouse or the generation of the power in the unit cell. Next function is it maintain or regulate the proper concentration of the calcium ions within the various compartments of the cell. Third is the energy transduction through the respiration. And next is the thermogenesis, that is the production of the temperature. Next is the endoplasmic reticulum. So endoplasmic reticulum are divided into two parts, that is it is having two types of endoplasmic reticulum. First is the rough endoplasmic reticulum and second is this smooth endoplasmic reticulum. So endoplasmic reticulum actually looks like what? It is a tubular or the flat vesicular structure in the cytoplasm. It is having a network-like structure which contains a cristae. So these are the cisternae which actually having a cisternal spaces. So the space inside the tubes is actually filled up with what? The endoplasmic matrix. So in this diagram, we can see a nuclear envelope which is consisting of the nuclear pore for the passage of the DNA materials. And it contains, these are the smooth endoplasmic reticulum and this dotted which contains the ribosomes are the rough endoplasmic reticulum. So these are having a particular structure, a folding-like structure, that is the cisternae. So this cisternae is having this space, creating a space which contains what? A endoplasmic matrix. So there are two types of smooth endoplasmic reticulum, that is smooth endoplasmic reticulum and rough endoplasmic reticulum. So in the diagram, we have first seen that in the smooth endoplasmic reticulum part, there is no ribosomes. And in this rough endoplasmic reticulum, there contains what? Ribosomes. So first difference is what? The smooth endoplasmic reticulum is not having ribosomes and this rough endoplasmic reticulum is having ribosomes. So smooth endoplasmic reticulum helps in the synthesis of what? The steroid hormones and the lipids, and the rough endoplasmic reticulum helps in the formation of proteins. And in the smooth endoplasmic reticulum, it actually having what? The lipid forming cells such as adipocytes, interstitial cells of testes, glycogen storing cells of liver, adrenal cortex cells, muscle cells, and leukocytes. And in the rough endoplasmic reticulum, it present what? A pancreatic acinar cell, goblet cells, which are in protein in nature, antibody producing plasma cells, Nissel's granules of the nerve cells, and many other cells, which contains what? Protein-like structure. Then comes the function. So function is firstly is the development or the generation of the proteins, which helps in the further generation of the DNA in inheritance. Then comes the protein segregation. And fourth is the unsaturation of the fatty acid, muscle contraction. That is, endoplasmic reticulum is commonly having another system, that is the sarcoplasmic reticulum in the muscle fibers. This sarcoplasmic reticulum actually helps in what? The passage of the calcium ion. So this calcium ion, when the concentration of the calcium ion increases inside the cell, what happens? It actually forms what? A depolarization. That is, a positive potential is created. This depolarization actually helps in what? In the muscle contraction. So after the mechanism is done, contraction mechanism is done, the sarcoplasmic reticulum again take back the calcium ion from the cell, inside the cell, and the membrane potential comes to the normal potential where depolarization is not existed and muscle gets relaxed. So these are the functions of the endoplasmic reticulum. Then comes the Golgi bodies. So these Golgi bodies are the collections of what? The membrane enclosed sacs composed of 4 or more stacked layers of the thin flat enclosed vessels lying near the side of the nucleus. So these Golgi bodies are actually having a stack like structure or coin like structure which is lying near where the nucleus. So it consists of what? Multiple discrete compartments. So this is having a multiple compartment. Here you can see there is a compartment like structure. So this compartment consists of what? Cis Golgi network, which is a Golgi stack, which is divided into the medial and the trans-sub compartments. And another is the trans Golgi network. So these are the trans Golgi network. So what is the function of the Golgi bodies? It helps in the packaging and the wrapping of the cell components inside the cell. Next function is the secretion of the granules. So whenever there is a vesicles present, it contains certain substance or the chemicals. It actually helps in what? The production or secretion of the granules. That is the membrane enclosed complexes which store, that is the hormone, enzymes in protein secreting cells. Then it forms what? The lysosome. It is a site of formation of what? Lysosomes. So that is the large irregular structure surrounded by the membrane which are present in the cytoplasm. So then it contains what? The carbohydrates which forms the glycoproteins, and these are the main or crucial substances which helps in the association or the development of the cells into the tissues. Then comes the lysosomes. So these lysosomes are actually in the diameter of 250 to 750 nanometer. So these are the irregular type of structure and it is a unit membrane. It is in the more acidic than the rest of the cytoplasm. So the pH of this lysosome is in more acidic in nature, and external bacteria as well as worn out the cell components and digested in them. So as it is having an acidic in nature pH, so this actually helps in what? The digestion of the bacterial elements or the worn out cells of the bacteria. So one thing I would like to mention that the lysosomes are also known as what? The suicidal bags. That is, it contains the enzyme, that is the lysozymes. So this lysozyme actually helps to digest the bacterial components or any cell debris outside the cell. So this interior is kept acidic near the pH of 5 by the action of the proton pump or the H plus or ATPase. Lysosomes are cell hydrolysis and they function best at the acidic pH. So in the lysosomes you can see there is a plasma membrane which is composed of what? The lipid bilayer and the glycosylated membrane transport protein. So function is what? It acts as a digestive system, that is a lysis of the cell, because the enzyme, the lysozyme present in the cell can digest the essentially all the macromolecules. It helps in the engulfing of the bacterial components of the cell in which they are located, engulf any harmful or exogenous substances like bacteria, and whenever there is a cell apoptosis, So this lysosomal enzyme causes what? The autolysis, that is a self-destruction. Autolysis means what? There is a self-destruction of the remaining substances. That's why lysosomes are known as what? The suicidal bags. Then comes the peroxisomes. So this peroxisome's diameter is 0.5 micrometer, right? So these peroxisomes can be formed by the help of budding of the endoplasmic reticulum or by the division. So it is having a lipid bilayer membrane that surrounds, which regulates what enters or exits the peroxisomes. Urate oxidate crystalline is the core of this peroxisome structure. Structure is similar to that of the lysosome but with a different composition. So peroxisome and lysosomes are having a similar like structure, but there is a difference in their composition. So it is having what? The urate oxidase crystalline core in the peroxisome. So it contains the oxidases that produces the hydrogen peroxide. It helps in catalysis, degrades the hydrogen peroxide to yield what? This water and oxygen. So this hydrogen peroxide degrades to form what? The water and the oxygen. Proteins are directed to the peroxisome by a unique signal sequence with the help of protein chaperones, that is peroxins. What is the function? It helps in the synthesis of plasmalogens, myelin sheet, synthesis of bile acids in liver, then the cholesterol synthesis in the animals, biosynthesis of the lipids, helps in photorespiration in plants, and hydrogen peroxide metabolism and detoxification. Then comes the cytoskeleton. So in this cell, the most essential part, that is the cytoskeleton, that is this systematic fibers that not only holds the structure of the cell but also permit the changes of the shape and movement of the cell. So the cytoskeleton is composed of mainly microtubules. So these are the microtubules. The intermediate filaments, so these are the intermediate filaments. Then microfilament along with the protein that actually holds or tie them together, right? Then comes the microtubules. So these are the long hollow structure which is having a diameter of 25 nanometer in diameter. It determines the shape of the cell, role in the contraction of the spindle, and the movement of the chromosomes and centrioles, as well as in the ciliary and flagellar motion. Then comes the intermediate filaments. The diameter is 18 to 14 nanometer and it is made up of subunits. They form a flexible scaffolding on the cell and helps in the resisting of the external pressure. If in their absence, the cell can rupture more easily, and when they are abnormal in human, blistering is common. Then comes the microfilaments. It is a long solid-like filaments which is having a diameter of 4 to 6 nanometer. They comprises of what? The contractile protein, actin, and are actually responsible for the cell motion. The function is actually, it helps in the locomotion of the chromosomes, then the movement of the cell, the process of secretion of the granules in the cell, movement of the proteins within the cell membrane. Then comes the most important part, that is the nucleus. So this nucleus actually contains what? The chromatins, RNAs, and nuclear proteins move freely in the aqueous solution. So nucleus is having a structure, that is nuclear envelope, nuclear pore, nucleolus, nucleoplasm, and the chromatin. So this nucleus is having what? A genetic material, and it actually helps in containing what? The nuclear functions. It is loosely organized matrix of the nuclear lamins extended from the nuclear lamina into the interior of the nucleus. So this laminas actually serves as the attachment of the chromatin and the organization of the proteins into the nuclear bodies. This chromatin actually helps in the nucleus forming a large loops of DNA, and specific regions of these loops are bound to the lamine matrix by the lamine binding proteins in the chromatin. Next come the nuclear envelope. So this envelope is situated outside the nucleolus or the nucleus. So it is having a nuclear pore complex. There is a two concentric membrane called the outer and the these are the outer membrane and the inner membrane. So this is the inner membrane, this is the outer membrane. So this outer membrane is continuous with the endoplasmic reticulum. So the space between the inner and the outer membrane is directly connected with the lumen of the endoplasmic reticulum. So this outer membrane and the inner membrane of the nuclear envelope is actually connected with a lumen-like structure of the endoplasmic reticulum. So it is having a permeable only to a small nonpolar molecules. So underlying the inner submembrane of the nucleus, there is a nuclear lamina, which is having a fibrous network-like structure that helps in holding the whole nucleus. Then comes the functions. So the nucleus actually allows what? The expression of the genes and regulated by the post-transcriptional mechanism, that is the splicing. The nuclear envelope provides novel opportunities for the control of the gene expression at the level of transcription, serves both as repository of genetic information and as the cells control the center. Then comes the summary. So this whole module actually consists of what? The cell and its function. So the function of the cell, the components of the cell, and what are the functions. So we can recapitulate in a short that the first is the plasma membrane, which actually helps in what? The passage of ions and the molecules. Then comes the cytosol. So it actually helps in what? The metabolism of the carbohydrates, lipids, and the amino acids. And next organelles is the mitochondria. It is a powerhouse or the energy production unit. Then endoplasmic reticulum, that is smooth endoplasmic reticulum, and the rough endoplasmic reticulum actually is the synthetic site or the synthesis site of the lipid, steroids, hormones, and the proteins. Then Golgi apparatus, it is the modification and sorting out of the proteins. Then the lysosome, the suicidal bag of the cell, which helps in the digestion of the cellular debris or the bacterial components. Next is the peroxisome, which helps in the utilization of the hydrogen peroxide, which can be degraded or catalysis to form what? The oxygen and water. Next is cytoskeleton, which is a frame-like structure of the cell that actually defines what? The morphology of the cell and the cell motility. And lastly, the important component, that is the nucleus, that is helps in the DNA synthesis and the repairment of the cell. So these are the references, and thank you.